We have a very exciting talk from an early adopter of SCP2019. And why was he an early adopter? He happens to direct the mass spec facility at Karolinskaya Institute in Sweden, and to work with Roman Zubarev, who was Bogdan's PhD advisor. So as we started developing SCP2019, Bogdan was in frequent communication with his PhD advisor, who was in frequent communication with Ákos, and therefore Ákos is ahead of the curve. And he's gonna tell us today whether he thinks that single-cell proteomics can be quantitative. I don't know for you, but I'm sitting on the edge of my chair. What if the answer is, I don't know. I'm just gonna listen to your talk and hope that you'll give the right answer.
Thank you for the introduction. It's very stimulating to be here, and I'm very happy to be a part of it, of all these discussions. I have learned a lot, and I try to make this fast and perhaps like a transition to the coffee break. Okay. So, I've been working in a proteomic co-facility more than 2 years. And so that means most of the time we deal with proteomic analysis at the bulk, with bulk material. We have a lot of cells, we have tissue samples, we have a lot of different questions coming from the biology, from different research groups, and many of them are very much interested in if we can move into the single cell proteomic level. So we have a great need. I can clearly confirm that. And we do as our best in our spare time to develop this SCP2019 methodology in our co-facility. And so we ask questions like how quantitative is single-cell proteomics? And I'm not sure if we have a clear answer to it, but I think I would rather leave it for the last slide. So there are a number of challenges, of course, related to the single-cell proteomics, and I'm just repeating a couple of things that you have already heard, and I can only confirm that, yes, there's a challenge. We deal with a very small amount of protein can be found in a single cell, and my estimate is like 200 picogram, which is, I think, in a good agreement with many of us, what we think about the cell can contain. And our sensitivity of modern mass spectrometry is not there, clearly. So if we translate these numbers, let's say calculating with an average protein molecular weight of 50 kilodalton, then we have about like 5 femtomole proteins in a single cell, right? And so it's nice to see to have like 3 billion of molecules that we can play with. But unfortunately, if we calculate with at least 10,000 proteins in a mammalian cell, then this number is melting down to 0.5 attomolar amount, which is, it's a challenge, right? So on the lower part, you can see how it is translated to in comparison with the bulk and the single cell proteins. And this is coming from the first nice paper. We need to face that we have a chance to detect and quantify the most abundant proteins. We lose a lot of edge already with the middle or middle high protein abundances. So in our single cell, not in a single cell, in a bulk routine analysis that we do in our proteome co-facility, we routinely work with and digest and label 25 microgram proteins. This is the level that we work with typically, and that is like 100,000 times more protein that one can obtain from a single cell. So one can choose a path, for instance, looking for the most sensitive instruments. There are always new technology coming up, and fantastic results are coming with new mass spec, like loading very low amount of proteins and routinely identify and quantify hundreds or if not thousands of proteins. We don't have these instruments. We have orbit traps. And we recently acquired a Lumos, we are very, very happy about this, but of course we have to deal with the limitations as well. So what we do is that we try to use and create a model, try to use this model to implement the methodology in a laboratory. We routinely use human cancer cell lines, a number of different cancer cell lines. In this presentation, I'm going to show you data obtained with archaeo cells. They are really tough cells, actually. So tough, I mean, they're difficult to get a good amount of proteins. But we treat these cells either with, as a control experiment with the DMSO or with different drugs. And these drugs are used at the IC50 level, and we measure the difference, how the proteome in these cells are disturbed upon drug treatment at different time points. In this presentation, I'm going to show you only data obtained after 48 hours treatment. So after 48 hours, these cancer cells are really changed the protein expression. And many of them already dying, but we are always analyzing the still living cells and discarding the floating dead cells in these experiments. So in this presentation, I'm going to show you data so archaeo cells treated with methotrexate, which has a well-defined target protein, increasing the expression level of the hydrofolate, the hydrofolate reductase, the DHFR. That you can. This is a slide that's showing our workflow. How do we do the experiment? How we prepare the samples? So either we take the control cells treated with DMSO or with a drug after 48 hours, and then we use FACS sorting. We isolate the single cells into a plate. This plate is a 96 plate low binding Eppendorf plate, which we found that works the best in our experiments. And instead of heating, we use a thaw method to extract the proteins. That works very well in our hands. And we have like 4 cycles of freeze thawing with liquid nitrogen. And after that, we digest the proteins in the plate format. After digestion, we directly apply then the TMT based labeling. So we have either 10 plaques or 11 plaques. We also have used 0 and duplex. I'm going to show you examples. And then we used our liquid chromatography MS data acquisition with the Lumos. In a lab, we usually use Proteome Discoverer for the data analysis, and I'm going to show you some results with it. So typically, when we analyze and compare the treated and control cells in these experiments and apply OPLS discriminant analysis to recognize these differences, we have a good separation of these cells between the control and treated cells. Yet, some cases we don't have a perfect separation. You can see that we have outliers. We have an overlapping region also in this analysis. I'm not saying that this is the most advanced statistical evaluation, but it gives us an impression that whether our sample preparation works well or we need to improve. And many times I think it's not only technical issue that we see this overlapping data, but it's also the biology. So we don't have a perfect separation of these treated cells. And most interestingly, for us, when we use the methotrexate treatment, we have the target protein, but many times we just don't see it in data. So how do we interpret? How do we access these problems? Whether there's a problem with the quantification or is there something else? Is that the biology? So maybe DHFR is not the best target, I would admit, because this is not the most abundant, not high abundant protein in our samples. But upon treatment, methotrexate treatment, the expression level increases, yet we don't see it in a single cell experiment. So this is an example of the single cell experiments. So for that reason, to address these questions, we have designed new experiments. And in this case, we didn't use single cell measurements. We have worked with bulk material, but the same thing. So we used archaeo cells treated either with DMSO or methotrexate. And we designed two different types of analysis. One is with carrier proteome as a classical, a more or less classical scope MS, but instead of using the carrier proteome channel as one of the TMT 10-plex or 11-plex, we have decided to work with TMT0. Advantage of using TMT0 is that we have a lower mass of the reagent compared to TMT 10-plex. And then we have the corresponding experiment designed without the carrier proteome. So you can see that we skipped the first two channels in the TMT 10-plex labeling, and hence we have 4 biological replicates, let's say, in each sample. And on top of that, we've been inspired by, as you have seen before today, by others' work. We did a dilution series, and we have injected either 100 nanogram, or sorry, 1,000 nanogram, 200 nanogram, 40 nanogram, 8 nanograms. That is corresponding to, in the first experimental design, when we have the presence of carrier proteome, we have in each channel, in each TMT channel, about 24 cell equivalent peptides, and about 5, 1, and 0.2 upon the dilution. Whereas the other design, when we have no carrier proteome at all, we have a lot higher level of protein in each channel. So, but we have a chance to compare two experiments parallel when we have either 25 or 5 cell equivalent in each. And then we have additionally one experiment with a more or less classical design when we have basically a single cell experiment, right? And I'm embarrassed to show you what is missing from this experimental design is the separation, and it's far from optimized. We have used only standard conditions that we typically apply for bulk analysis. So that was also part of the design. And also lack of time that we haven't looked for optimized conditions for the separation, which I think one of the key elements that we need to implement in order to get higher coverage and better quantification. So we use this easy column with a nanotrap and a 2 hours gradient, very straightforward conditions. So let's start with experiments without the carrier proteome. So this is really like what we typically see in a bulk experiment. And if you look at the figures, first of all, yeah, we have the mass spec conditions. These are very typical conditions that we use, except for the collision energy. It's slightly higher that we typically use for the TMT-based bulk experiment. But looking at the proteins, we could actually see and quantify more than 1,000 proteins, right, in experiment when we had 40 nanogram injected on a system. I was very happy with this data, actually. And looking at, furthermore, the number of spectra collected, including also the number of PSMs, it is very promising data. We are talking about a low amount of peptides injected on a chromatographic system and analyzed by mass spec. So it is, I think, very promising, yet we don't see basically very, very low levels of proteins, very few proteins when we inject 8 nanogram on column, which is not that impressive. And 8 nanogram is, like we have discussed before, like 40, maybe 40, 50 cell equivalent, right? Sorry, then it's loading too slow. So what is interesting to see is that we have calculated, we have 4 biological replicates in the design. We have calculated the fold changes at both the protein and also PSM levels. What we have seen is that injecting 1 microgram on column, we could easily see the DHFR, target protein, and it's clearly upregulated in our experiments. This can be seen also when we inject less, like 200 nanograms on a column, but yeah, it's just disappearing when we work with 40 nanogram. And that's perfectly in line what we have seen in single cell experiments. So we cannot expect actually to pop up proteins which are not highly abundant in our experiments, even though we inject like 40 nanogram on column. The PSM level quantification, so fold changes, shows us that we have a number of peptides originating from the DHFR. So that's corresponding well to the protein fold changes. And they are also showing upregulations. This is pretty consistent data, I would say. And we have surprisingly good results with even the lowest 8 nanogram injected on column. I mean, 100 proteins is not much, not many, but I mean, it still could be useful. So when we compare then the fold changes, so we do the correlation, Pearson correlation between the low amount of sample injected in column with the 1 microgram, the higher level, we can see that the correlation is rather high. We have very good correlation even with the lowest amount injected on a column when we compare with the bulk experiment. This is exactly what we want to see, and this correlation is significant, not only at the protein level, but at a PSM level. But you can see that at PSM level we have less consistent data. We have lower correlation as well, and so that is perhaps understandable that some of the peptides we have seen in the PSM data can sum up and improve the quantification of the proteins when they are considered. And we haven't filtered anything. We have included as many PSMs as possible, and we accepted also protein identification by 1 peptide only. So going to the second method that we have investigated, as I mentioned, we used the TMT0 to label the 200 carrier proteome, and we utilized then the difference between the reagents, so the mass, the residue modification difference between the 0 and the 10-plex, which is like 5 Dalton, and then we used the Lumos with the isolation offset of 5 Dalton to acquire data. And the idea was here is to use either the orbit trap or the ion trap, and it turned out to be the ion trap was a better choice for precursor selection and generating MS2 spectra for identification purpose, and trigger the quantification, the MS2, another MS2 event used for quantification with an offset of 5 Dalton. And this design is it has advantage that we can separately tune of these 2 events, but it has inherited problems. I'm going to come back to that problem. Looking at the data, we have, it's just like, it shows that the difference between acquiring data in a first MS2 event in an ion trap or the orbit trap, we found that the ion trap was more sensitive as expected, and we got pretty good identification data generated with it. So we use lower energy for the fragmentation in this event compared to the quantification event, now the MS2 event. And we had also the chance, of course, to play with the injection time. This is then, it's not optimal, but this is what we typically used before successfully in single-cell experiments. So again, looking at comparing the different amount of samples injected on a column and looking at the data, we have found that injecting 40 nanogram on column, which is corresponding to our single-cell design by scope MS, then we have more than 500 proteins identified, which is, I think, rather good. And also, we have high number of spectra acquired. But then the problem with this design, this method, is that we spend too much time. We split the time between the two MS2 events. So we are not able to generate as many MS2 quantification spectrum. And that's another problem that using the Proteome Discoverer doesn't allow me to match directly the identification information with the quantification. So yeah, I have to do everything manually. So maybe I think I should turn to MaxQuant to. No, I mean I learn to improve this point of the analysis and then improve the identification. So looking at the fold changes, we have slightly different data, and interestingly, the DHFR doesn't seem to be coming up at the right side of the plot, which is, I think, again related to the identification. So we may have a disconnection between the quantification spectra and the identification spectra, and unfortunately, we don't see any proteins or PSMs in the lowest amount of injected, which is, I have to remind you, it's like 0.2 cell equivalent in each channel. So maybe that's acceptable. Again, comparing the lowest amount of samples injected with 1 microgram, like a bulk experiment, we still have very good correlation, and this is significantly a positive correlation. That's what we would like to see. And based on this, I would dare to say that we have a good quantification. Yet, we have to, I think, further investigate and validate this statement, I would say. So when we compare the two different methods, I would say that at this level of detection, so when we detect our single cells based on this scope MS method, we need to have the carrier proteome. Our instrumentation doesn't allow us to go deeper without the carrier proteome, and so when we look at our single cell data, we need this effect coming from the carrier proteome. That's my conclusion. And coming to almost the last slide, so I dare to plot also and estimate how does it look like, how does it compare in these two different methods. So what you can see here is the number of quantified proteins against the number of calculated cells per TMT templex channel, and this is not a fitting. This is no. I just draw a line to indicate what is my desire. So I want to routinely quantify at least 1,000 proteins in each experiment. That would be a deliverable to our customers, and it seems that without the method without the carrier proteome, and also the method with the carrier proteome shows something similar, namely that in order to reach or meet this requirement, I would need at least like around 200 cells, I would say. Here we have like this plot indicates a 17 would be a good number here. So that's translated to a 10-plex experiment to 170 cells, but of course it's a very rough estimation. And here with the carrier proteome method, we see that we already reached that level after 1-2 cells in each cell, each TMT channel. So this is my final conclusion. And also the quant spectra is something similar, as you can see in the smaller side on the right side. So in conclusion, I would say that we have the current methodology gives us quantitative data, and I'm very pleased with that because there's a fuel for us to continue our development. And it seems like we need an order of magnitude boosting this effect of the carrier proteome in order to meet this 1000 protein quantification as our goal indicates. And that's what we have, like 200 cells approximately. And I'm not continue with this, and I would like to just mention that either we continue with the carrier proteome approach and we develop the methods, or we use something else without maybe data independent acquisition. That would be a nice option to see how it develops. But I think we are getting there. And with that, I would like to also thank our funding bodies and Mari Stolberg for technical assistance. And I'd like to also thank you for your attention.
Thank you. I have a burning question, so I'm going to ask first, and then I'm going to let Claudia ask. The obvious question that I had in my mind during most of your talk is, why didn't you target DHFR? If you know what is your target of interest, just put it on an inclusion list and accumulate it for as long as it takes, and then you're going to see it with much higher probability.
Yeah, we don't want to get it granted like that. I mean, we want to see it without targeting directly the target protein, let's sort of say. That would be, I think, more convincing for...
Not in terms of quantitation. In terms of quantitation, if you were to target it, your quantitation is going to be just as reliable.
Yeah, or maybe it is just a dream that we see, what we would like to see, because many times that even the bulk analysis doesn't give you that power to obtain reliable data with your target proteins. But yeah.
All right.
Sure.
Thank you.
Yeah, thank you.
Question. Who has the other mic? Is it great? Okay.
Very interesting. When you try to infer your quantification and your identification from your offset, have you ever looked at your fragment ions if 100% actually overlap, or do you see differences in there?
It's a very, very good question. I thank you for that. I think the greatest issue with that approach, although we separate the precursor very well, but with the 5 Dalton shift in our second MS event selection, we also co-isolate other precursors that can interfere with our data. And unfortunately, I can see too often the 126 coming from the TMT0, even in the quantitative data, which indicates that this is the case.
Bogdan has a question here.
Yeah, it's really nice to see that they actually SCP2019 started working in Europe. That's a great step.
Yeah, thank you.
And the question related to that, I do actually expect that when you isolate empty space plus 5 Dalton, then it still will be co-isolation, that it's just not visible to you. That's why I think when you look into this PSM correlation between 1 microgram and 40 nanogram, I think that you need to actually look in some filtering, because you did mention you did no filtering, it just all the data right there. So I think you need to be paid a little bit more attention to look at those outliers. I bet those that have a huge co-isolation of something has nothing to do with your ions, respectively. They are actually lower artificially your correlation coefficients.
I fully agree with you. And of course, my current plan, what is outlined during this nice discussion, is that I'm going to move on to the max quant and learn how to tune that investigation. Yeah.
So I guess my question opens up the floor more widely. Does either Proteome Discover or Max Quant allow for researching the isolation window with an offset to find potential contaminating precursors? I don't know that Proteome Discover has that baked in.
Jorgen's answer is MaxQuant is not looking with an offset, doesn't have that feature.
That's a great talk. I have two questions. And the first one is, did you did MS3 on Lumos?
Yeah.
So compared to MS2,
I
even lower signal.
No, it's a no-go at the moment. I think we don't have as many ions to play with. That's a problem.
Yeah, definitely. And the second question is, did you ever correlated the result from your two settings? Exactly the without control and with control and without control. You have exactly same setting with some chemicals.
Mm-hmm.
So when you have a lot of correlation. Did you ever tested this correlation?
Yeah, it's a good question. I haven't done that, but it's a good suggestion. Thank you. I'm going to do that.